So today I'm going to tell you about some of my lab's work on the presynaptic nerve terminal. I'm clearly the token presynaptic talk for the day. We've heard a lot about the postsynaptic cell and how the regulated trafficking of glutamate receptors into and out of the synapse can mediate forms of plasticity like LTD and LTP. Today I want to focus your attention a little bit on the presynaptic side of the synapse and tell you about some of our work there. As you know, action potentials invade the presynaptic nerve terminal. They cause the opening of voltage-gated calcium channels. Synaptic vesicles then fuse with the membrane, releasing neurotransmitter contents on the postsynaptic cell. That gives rise to an evoked response, where many synaptic vesicles can fuse at synapses. The synapses can also undergo what's called spontaneous release, where individual synaptic vesicles can fuse in the absence of any stimulation. This has historically been thought of as noise in the system because it's not enough to drive action potential propagation in the postsynaptic cell usually. But today I'm going to tell you a little bit more about spontaneous release and some of its biological roles. But I want to mostly focus my attention on how the presynaptic cell releases neurotransmitter release and how release probability is actually set. So one of the tools that we recently developed in the lab is sort of quantum resolution optical imaging, where we can watch single synaptic vesicles fuse at individual release sites. And that allows us to really determine precisely how individual active zones are performing and how their dynamics can change. You've been watching spontaneous release going on here in real time at the Drosophila neuromuscular junction. So the model synapse that we study is the NMJ of the fruit fly. It's a glutamatergic synapse. Motor neurons will exit the brain. They will form stereotypical synapses onto these muscle fibers. As the animal grows throughout the larval stages over about a week, these synapses grow dramatically. The motor neuron will increase its branching and the number of active zones that are placed here as the muscle grows in order to maintain contraction. This is all regulated by neural activity, so it's a really pretty synapse to think about how structural plasticity is regulated by neural activity in developing simplified synapse. We can also watch this biology in live animals, so we can actually watch synaptic growth in vivo. So here you're watching a presynaptic arbor budding out a new synaptic bouton. This occurs on the order of 10 to 20 seconds, so it's very quick, very rapid. So this is what the synapse really looks like, and I'm going to sort of focus our discussion here for just a minute to tell you about the method that we've been working on. So here's the axon coming into the muscle fiber. It branches over the muscle surface, and it puts on the muscle surface anywhere from 200 to 400 individual active zones. These individual active zones in the fruit fly are called T-bars. So here's an EM picture of a slice through one of these boutons. This is a swelling of the axon where these individual active zones are placed. These little umbrella-like structures are called T-bars. Here's a magnified view of the T-bar. The T-bars clusters the presynaptic calcium channel, and it clusters synaptic vesicles, and it's the site where these synaptic vesicles will undergo fusion with the plasma membrane. As you can see, each of these individual active zones, which are marked here by an antibody against the Drosophila Cas homolog called BRP, is opposed by an individual cluster of glutamate receptors, and each of these active zones is spatially segregated with each other. And fly glutamate receptors can actually flux calcium, and that's allowed us to tether postsynaptic calcium indicators to the postsynaptic compartment and watch calcium influx following single synaptic vesicle fusion events. Now, historically, we have characterized synaptic transmission in the system as many of you do. We put electrodes into the postsynaptic cell and watch current flow. When we stimulate this motor axon, we can get anywhere from 10 to 20 or more synaptic vesicles fusing, but we have no idea where those synaptic vesicles are originating from. And using this method that I've briefly mentioned, where we can tether postsynaptic G-camps, we can actually visualize where synaptic vesicle is fusing, and we can also do physiology at the same time. And basically, we can see these flashing events whenever a synaptic vesicle fuses, and we record a mini at the same time. Here you can see 10 seconds of imaging that we've collapsed into a single stack, just showing 10 individual spontaneous fusion events that have happened at this synapse. So what does this look like in vivo? So here is a neuromuscular connection, the axon coming in. This is watching spontaneous release. So we cut the axons from the brain and put it into a suction electrode so we can control the firing rate of these neurons. You can see spontaneous release happening across this population of about 200 to 300 individual release sites. When we fire an action potential, we get a very different response. So now we still have spontaneous release going on in the background, but now we get the synchronized fusion of synaptic vesicles across numerous active zones. And by following where these synaptic vesicles are fusing at individual release sites, we can determine a variety of important parameters. So here you see two neuromuscular junctions that we've reconstructed. All the little white dots here are the individual active zones. This is one NMJ connection. This is another. On the left, you're seeing spontaneous release. On the right, evoked release. These are ongoing at the same time. We've just artificially separated them. We can do that because the evoked events are time-locked to the action potential. Okay? So we can basically watch these synapses work in real time, and we can follow release events occurring over individual active zones. So that allows us to determine the rate of release for both spontaneous events and release probability for individual evoked events. We can determine how these active zones set their individual release probability and how those release probabilities might change with, for example, plasticity paradigms. We don't have in this system, for example, branch point failure. So if you look at these release events during an action potential, you see them all over the arbor that are occurring. We don't really see, for example, that events are only happening at one branch of the arbor and not another, so that we don't have branch point failure in the system. So here's some of the questions that we wanted to ask with this approach. Going into it, I've been studying vesicle fusion pathways for a long time, and I had assumed that every single active zone would basically have both modes of fusion, spontaneous synaptic vesicle release and evoked vesicle release. That was my assumption going in, and I'll show you, in fact, that that's not true. We also sort of thought of going in that spontaneous and evoked release probability would probably be relatively constant over all of the active zones, that the motor neuron would place a set of about 200 active zones on this muscle fiber, and they would all be about basically the same flavor, and that turns out not to be true either. And so that's led us to looking at another number of other important questions that the system has allowed us to address. One is actually what determines release probability. As I'll show you, neighboring active zones can have release probabilities that differ by over 100-fold. So what makes a very strong high P active zone versus a very weak low P active zone? And what makes, for example, a spontaneous only active zone versus one that has both modes of fusion? And finally, ultimately, what we want to do is begin to engage plasticity mechanisms at the synapse and ask how does changes in presynaptic release properties contribute to various forms of plasticity, as we've heard about for regulated changes in glutamate receptor trafficking in the postsynaptic cell. So we've had a special interest in spontaneous release for a long time in the lab. We've known for a while that spontaneous release rates can be modified by activity. I'll show you some of that biology. And they also play important roles in sort of structural plasticity at the synapse. So we were interested in looking at spontaneous release and asking whether there was any regulated pattern to release events that were going on. And what you're looking at is an arbor here. These are the individual active zones. And basically, you're looking at a movie that's been sped up, has 5 minutes of imaging at this synapse. And we're just exaggerating the decay time constant of these spontaneous fusion events just to allow you to follow them spatially over the entire arbor. And what we find is these events appear to be relatively random. We don't see coordinated fusion events at individual boutons, for example. They seem to be, in fact, relatively random over the entire arbor. So there's no obvious pattern that we could pick out in the absence of any stimulation. They're also relatively stable, so if we image the release probability, either for spontaneous or for evoked, we let the preparation rest and we come back and remap release probability, we typically find that the release rates change very little. The release probability seems to be inherent to the active zone. If we do see any changes, they're very small. About 76% of the active zone events, release probabilities didn't change at all. And the ones that did are relatively subtle. So some of the rules that we've been able to pick out for how individual active zones work in terms of spontaneous release are shown here. The average rate that we find for a single synaptic vesicle fusion event in an individual active zone is a fusion event about once every 80 seconds. So for those of you that care about homeostatic plasticity mechanisms and are interested in how spontaneous fusion may regulate that, basically you can think about a time clock, potentially a postsynaptic calcium-binding protein that works on a time scale, something like this. We were also sort of interested in following whether once a single synaptic vesicle fuses in an active zone, is there a delay in which another synaptic vesicle can fuse? Is there some type of required docking and priming reaction that sort of worked as sort of a little railroad track that potentially once a fusion event happened would require some type of time reset? We don't see that. We can see that a single active zone can have another fusion event at that active zone as fast as we can optically measure it. So there doesn't appear to be some required repriming event going on at these synapses. And again, as I mentioned before, release probability is highly variable across the active zone. And indeed, some active zones don't show spontaneous release at all during our imaging sessions. So the big question that we were sort of interested in was, does every active zone behave the same? Does it have both spontaneous and evoked release? And in fact, the majority of the active zones do. So here you see an individual active zone showing a spontaneous event, spontaneous, and then it shows an action potential, which you can see by other active zones also releasing synaptic vesicles. So most of the active zones, about 70% or so, are these mixed mode active zones where we see both modes of fusion. At these active zones, we can also address another question we were interested in, and that is whether there was multi-quantal release going on at single active zones. Can one of these single active zones in the fly release multiple vesicles? And at the mixed mode active zones, we can basically measure our delta F signal for the spontaneous events, which are single vesicle fusion events, and then for the evoked responses. And for the most part, we find that a single synaptic vesicle will fuse in an individual active zone. The events are uniquantal, and you get the evoked response because many active zones now participate in the evoked response. If you go to really high calcium concentrations, you can get more than one vesicle to fuse at these active zones. So I don't think it's an absolute requirement that, absolute rule that they can't, but generally release probability is so low that they don't. Okay? So here's an NMJ that we've reconstructed release probability for spontaneous fusion and for evoked fusion. And as you can see, release probability is highly variable. So you'll sort of see these throughout the talk. These darker colors, the redder colors, are sort of high P active zones. The bluer are more low P. And if you look over in Arbor, what you find is that a small subset of the active zones, these here in the darker colors, are very high P, whereas most of the active zones at the NMJ are very low P. Low P on average is about 0.04, which is somewhat similar to what you find at a hippocampal synapse, which is about, I think, 0.05 or so. So for 100 action potentials, only 5 synaptic vesicles would have fused over those 100 action potentials. And these active zones can be right next to each other. So a high P evoked active zone can sit right next to a low P active zone. So this seems to be regulated at an individual active zone level. If we sort of compare spontaneous release rates, so the number of spontaneous events that are occurring in active zones, to the evoked release map, we actually see that these are quite distinct. We sort of thought, or certainly I thought going in, that an active zone that's good at releasing a vesicle would be good at releasing either a spontaneous or an evoked vesicle, and that turns out not to be true. So active zones that have very high probability for evoked release, it has no bearing on whether it's going to be a lower or high probability release spontaneous sites. The same is true for these high spontaneous release sites. It really has no bearing on their rate of their release probability for evoked response. So these two modes of fusion appear to be distinctly regulated at individual active zones. The other real surprise that we found is that a subset of these active zones, in fact, didn't participate in an evoked fusion at all. So a typical imaging session is about 10 minutes. We're stimulating at about 0.3 hertz or so. And here you can see in blue are sites where, in fact, these active zones never, ever released an evoked event. So we call these spontaneous only active zones. And it makes up about 20% of the population of active zones in our system. So basically, there's a dedicated pool of glutamate receptors at 20% of the active zones that only listen to spontaneous events. There's been a big mystery in the field about how a postsynaptic cell can tell the difference between an evoked fusion event and a spontaneous fusion event. And at least in our system, one mechanism might be that, in fact, some of these events are segregated, that there's a population that is spontaneous only. So we have both high P and low P active zones, and then we have these spontaneous only active zones. If we raise the calcium, we see what you see at every synapse. Release is incredibly sensitive to external calcium levels. So as you raise the calcium levels, you can increase release probability across the entire population, and you can increase release probability from this 5 to 10% of high P synapses as well. And very high calcium, you can actually drive these, the top 5% of active zones to release probability of 1. So they will release a vesicle to every single action potential. So this is, okay. So this is, of course, a mystery for us is really how you set release probability at individual active zones. And of course, in terms of thinking about plasticity, is this a dynamic property of individual release sites? Can they be modified up or down? Might you recruit spontaneous only sites into evoked sites with some type of plasticity? Those types of questions. So that's something that we're very interested in addressing. And there's sort of several potential mechanisms that we thought about in terms of what might set release probability. One idea is we know very little about how individual synaptic vesicles are recycled per active zone. We don't really know much about how they mix between individual active zones. These release sites in the fly are segregated over about 700 to 800 nanometers, so they're quite far apart. Over a long time, certainly vesicles from one active zone can mix with another, but on a short time scale, it's unclear. So we thought maybe these high P active zones could be an instance, for example, that they have a pool of synaptic vesicles that are unique from the low P. Maybe these have experienced some type of a phosphorylation event. For example, the calcium sensor Synaptotagmin might be phosphorylated to make it a higher affinity calcium sensor, something like that. So we're very interested in testing that model, whether in fact there might be something unique about the synaptic vesicle proteins. As a geneticist, the way we think about this is that the major determinant of release probability, if we take it away, all the active zones should go blue. We should basically lose this population of really high P active zones. The more obvious explanation is potentially that something is unique about the active zone itself. For example, the number of calcium channels might define a high P active zone versus a low P active zone, and might in fact contribute to these spontaneous only active zones. Perhaps they lack voltage-gated calcium channels altogether, and therefore couldn't respond to evoked response because they lack local calcium influx. Alternatively, something about the active zone structure itself, independent of the calcium channels, that promotes synaptic vesicle docking or priming might be a key determinant here. So we set about to test these molecular models in the lab, and this is sort of work in progress, so I'll tell you about that. The first thing we did is actually begin to examine the release machinery itself, and particularly our favorite molecule, Synaptotagmin, which Tom's lab actually cloned back in the day, and I've been working on for 25 years, which is sort of depressing. You've been working on it even longer, which is perhaps more depressing. But the way these synaptic vesicles fuse at synapses is through a regulated molecular machine that's present in all cells, the snare complex. This brings these two lipid bilayers together, which don't like normally to be around each other. And neurons have evolved proteins that basically control the assembly of this snare complex and make it sensitive to calcium. And so one of the key factors that we've been studying is the synchronous calcium sensor synaptotagmin. This is a synaptic vesicle protein. It has two calcium binding C2 domains. Upon calcium influx, it dives into lipid bilayers and may engage the fusion machine itself as well. Whether you knock this out in flies or in mice, I think essentially the phenotypes are the same. This is a wild-type animal that has normal synaptotagmin levels. When you stimulate it, a number of synaptic vesicles fuse. This is 10 to 15 synaptic vesicles fusing at the embryonic synapse, some work done by Moto Yoshihara when he was in the lab. If you take away synaptotagmin, what you see is that you no longer get this synchronous phase of release. So here's the action potential. You lose all these synchronous vesicle fusion events, and instead you can still get fusion events that happen. They're much fewer in number, and they're asynchronous from the action potential. So if you just look at the latency, typically in a wild-type synapse, all the vesicles fuse within a few milliseconds. Synaptotagmin mutants, you can have vesicles fusing out hundreds of milliseconds later. This is called the asynchronous release phase of release, and there's debate about what calcium sensor, in fact, may mediate this. But clearly, synaptotagmin drives this fast phase of release. You don't see this phase of release at wild-type synapses, and synaptotagmin plays a role in suppressing that biology as well. We see this with optical imaging as well. So if we optically image release in a synaptotagmin null mutant, unlike a wild-type animal, when you stimulate, you see this nice synchronous fusion of vesicles that can be detected across many active zones. When you stimulate in a synaptotagmin mutation, everything, as we see physiologically, is completely asynchronous. We get no synchronous fusion events of synchronous vesicle fusion events. However, even in that background, if we look for either spontaneous release or we stimulate to get a lot of fusion, for example, through giving trains of release, we still find that there are preserved active zones that have much higher release probability than their neighbors. Although release probability goes down for all the active zones, there is still this protected pool of 5 to 10% of the active zones that have a much higher release P than the majority of them. And that is sort of arguing that there's something potentially unique about the active zone rather than the synaptic vesicle that may sort of shape what release probability is. So therefore, we began to ask the question that was sort of more potentially obvious: is this simply a factor involving calcium channel density? If you have more calcium channels at high P sites, you would potentially get more calcium influx, and therefore that might lead to a much stronger active zone, a higher release P. So the first thing we did is to look at the calcium channel distribution at Drosophila active zones. And this is a little bit of a challenge because we don't have good antibodies in the field. We've made, I don't know, about 10 at last count that don't really work well. So we basically have to use overexpressed exogenously tagged channels. And these are tagged with GFP. So this is Drosophila has one N-type calcium channel called cacophony that mediates all presynaptic calcium influx. So there's only one of these players here. And if you look at its distribution, we in fact find that it is heterogeneous. We can see active zones where there's a lot of CAC overexpressed signal and active zones where there's very little. This is a marker for the active zone, so we can identify the active zones. These are the glutamate receptor fields. And there are, in fact, sites where we see active zones that lack calcium channels altogether, but they have glutamate receptors. And about 10% of the active zones show this mode that we can't detect overexpressed calcium channels. And we see about 20% of the synapses are spontaneous only. So this may play a role in the spontaneous only pool that I told you about. However, what you really want to look at is, in fact, calcium influx. Calcium channels are the subject of all sorts of modulatory effects. So just examining the density of calcium channels is really not what you want to do. What we really wanted to do was be able to measure calcium influx presynaptically at a single active zone, find what that calcium influx was, and then ask whether a vesicle fused at that site or not. The problem is we're using GCamps on both the pre- and postsynaptic side of the synapse, so that makes it very difficult. So the first thing we had to do is actually figure out a way to measure calcium influx at individual release sites. Our approach at the neuromuscular junction postsynaptic was just to take GCamps and myristylate them, and that packed them on the postsynaptic membrane. And because the glutamate receptors are clustered in fields opposite each active zone, we didn't need specificity of targeting. The specificity came from the calcium influx. For the presynaptic side, that didn't work, so we spent some time figuring out how to actually generate reagents to measure individual active zone calcium channel. And what finally worked is by tagging BRP, which is the active zone component, directly to GCamp. That allows us now to follow, during stimulation, the influx of calcium at these individual active zones. And indeed, what we find is that some active zones, and these are just multiple stimulation trials showing stereotypical calcium influxes at these active zones. Some are high fluxing sites, some are low fluxing sites. So there is not only heterogeneous distribution of the calcium channels themselves, but also in the amount of calcium influx coming in presynaptically. And we can correlate those two. So we also have CAC transgenes labeled with red in cherry. So we can correlate the intensity of calcium channels in an active zone with the amount of a BRP GCamp calcium influx signal that we see. And in fact, these are tightly correlated. So essentially, the influx of calcium tightly tracks the density of calcium channels that are there. So, and that allowed us basically now to use the red calcium transgenic animals and then do our postsynaptic GCamp imaging and ask, as we saw for calcium influx per calcium channel density, is that correlated to release probability? Again, here you see that correlation. And what we find is that we do see a correlation, so here you can see it, but it's not nearly as tight as what we saw for calcium channel density. It's even less tight for spontaneous release. So for spontaneous release, there's very little correlation at all between the spontaneous release rate and the density of calcium channels in an active zone. So certainly calcium channel density does play a role. The more calcium channels you have, typically the higher evoked release probability you have, but it's not absolute. For example, you can have active zones that have essentially the same amount of calcium influx that can differ in release probability over a range of 10 to 20-fold. So it's not the only determinant. It appears to be an important player, but it's not the only determinant. So factors that regulate either the opening of the calcium channel or their density at active zones influence release probability, but they're not the only player. So what else might be going on, and this is sort of where we are now, is a variety of other proteins that are found at active zones that play in roles outside of just clustering calcium channels. There's a variety of proteins involved in vesicle fusion. There are proteins involved in priming and docking. And so as a next stage, we're interested in beginning to look in these various mutants of the priming and docking proteins and asking how these manipulations, which might alter actually synaptic vesicle number per active zone, might they contribute together with calcium channel density in shaping release probability. So that's sort of where we are in beginning to define the basic properties of release probability. And then I just want to briefly sort of show you some of the work that we're doing to look at plasticity mechanisms. Is in fact presynaptic release a plastic mechanism that can be altered at the synapse? And one of the forms of plasticity that's very robust in Drosophila, back when we were trying to, when Moto was in the lab, look at LTP-type mechanisms. If you give fly motor neurons LTP-like stimuli, what you actually see is they have a dramatic increase in spontaneous release rates. This is transient. It lasts several minutes, and it goes away. So this is the most dominant effect of high-frequency stimulation of a fly axon. Here you can sort of see a low-frequency spontaneous release rate and a high frequency. We've done optical imaging on this biology. When we give a high-frequency stimulation, we see an increase in mini-frequency across essentially most all the active zones and a recruitment of sites that were previously silent. So this increase in spontaneous release appears to be playing out over many active zones. And I don't have time to tell you about the biology, but we've worked a lot on this in the lab, and it has some similarities to mammalian forms of plasticity like LTP. It requires calcium influx into the postsynaptic compartment through these glutamate receptors. It actually requires a novel isoform of the Synaptotagmin family called Synaptotagmin 4, which binds calcium and mediates the release of a retrograde signal. That engages PKA, and PKA is in fact the critical factor that increases mini-frequency. And so very briefly, I just want to tell you about our work on trying to figure out what it is about PKA activity that drives this change in release from basically regulated evoked fusion to switching the synapse transiently to the spontaneous release mode. You can see that here. So here's the time course of this biology. Happens over several minutes, and it goes back to baseline. It's absent in the SIT4 mutant, and it requires pKa. You can either use mutations in pKa or a pharmacological inhibitor, and you completely block this type of biology. And basically, what we found is this gating of spontaneous release works through a protein called complexin. So the snare complex, which regulates neurotransmitter release, has another binding partner, again, that Tom identified in his lab, called complexin. Complexin binds the snare complex in a one-to-one fashion. There are different models about how it actually works. It plays a role in both driving evoked release and in clamping spontaneous fusion. My favorite model is that it actually controls the zippering of the snares. The complexin competes for zippering of the termini of the snare complex and basically prevents the vesicle from fully fusing. Upon a calcium influx and synaptotagmin activation, this clamp is relieved and you can progress to fusion. If you take away complexin, so we've made mutations in fly complexin, you get this huge increase in spontaneous release. In fact, it looks like this. This is a Christmas tree type effect. These axons aren't being stimulated. This is the complexin null mutant. We see this dramatic enhancement of spontaneous release over the entire arbor that has, in fact, some similarities to what you see with activity. So we sort of had this model that potentially complexin could function as the regulated clamp for spontaneous release, such that at a normal synapse, spontaneous release would be a low rate because complexin is engaging the snares and preventing it from fusing. During this high-frequency stimulation, we build up postsynaptic calcium. It engages a retrograde signal that activates pKa. It phosphorylates complexin, and that temporarily unclamps release for several minutes. And we get this large increase in spontaneous release before the protein is dephosphorylated and the system goes back to normal. And so one of the big questions is why the hell you would actually want to do this? Why does the system matter that you're having an increase in spontaneous release? Well, we think that what this does is basically it allows the memory, or the synaptic tagging, if you will, of a strong stimulus that allows postsynaptic calcium to remain elevated for several minutes. Because the minis are engaging and activating glutamate receptors, we get continual calcium influx here. And we think that prolongation of the calcium signal, and I'll show you very quickly some results that indicate that's true, engages growth mechanisms at the synapse and helps to shape how activity can drive structural changes at the synapse. So we mapped out the complexin, the PK phosphorylation sites in complexin. It's a small protein. It only had 5 potential serine-threonine residues. We individually mapped this out. We found that serine 126 of one of the splice variants is the site of PK phosphorylation. Mutation of that residue gets rid of most of the phosphorylation of the protein. So we made phosphomimetic and phosphoincompetent versions of that. The phosphomimetic version actually increases spontaneous release rates on its own when we replace the wild-type allele, whereas the phosphoincompetent version has normal spontaneous fusion rates, but it can't respond to this high-frequency stimulation. In the phosphomimetic version, although it increases mini rate, it cannot respond to this as well. So this type of plasticity is occluded when we mimic phosphorylation of this protein. We have lots of data implicating spontaneous release and structural plasticity. I won't show you that now. I'll just show you this one figure of one of the assays that we use in the lab. If you increase the activity of the motor neuron, you can do this through many different ways. What you find is that the number of boutons that are formed onto the muscle surface can dramatically increase. This is well known in the fly field. An easy way to do that is just to shift the temperature of the animal it's grown at. You get more motor neuron firing and more movement, and the animal grows dramatically. In the complexin mutant, if we put the wild-type complexin transgene in there, it works perfectly normal. We put the phosphoincompetent version, this form of structural plasticity is completely gone. And if we put the phosphomimetic version, this animal actually grows more synapses to begin with, and it shows no increase with activity at all. The same is true if we overexpress the constitutively active pKa in the presynaptic terminal. We can promote synaptic growth, and we do that in the background of the phosphoincompetent version. We can no longer promote synaptic growth in that background. So I'll end there sort of with the idea that clearly the presynaptic terminal has very dynamic mechanisms for changing release mode, and we're very interested in whether release probability might also be regulated. And I think future studies are clearly going to be focused on what it is that sets release probability. How do you make an active zone that can sit next to its neighbor and yet be 50-fold stronger? Clearly calcium channels play a role, but they're not the only story. So that's sort of an area that we're very interested in the lab. So the work that I told you about was done originally by Jan Mellem and picked up by Yulia Agwarjanova in the lab. And then the work on complexin is done by Richard Cho, who's currently going on the job market for any of you looking for excellent Drosophila neurophysiologists. So with that, I'll thank you. Take any questions. So I wouldn't call this LTP at all. I would call this an activity-dependent change in presynaptic release properties. So I think morphing it, it's a short-term plasticity change. Lasts over about 3 to 4 minutes. There's no, I would say, no real LTP-like phenomenon that goes on at this neuromuscular synapse. There may be in the CNS where those synapses are more in tune to short-term acute changes, whereas the neuromuscular junction is really growing over a slower time course. So I would say NMDA-type dependent plasticity has not yet been found in the fly, but certainly the fly has plenty of forms of plasticity, learning, and memory paradigms out there. So there are going to be molecular mechanisms that mediate that. And whether they're pre or post is unknown at this point.
So the data that you have on the complexin are really interesting, Troy. And I was wondering, as you probably know, you can dissect the molecule based on different domains that are involved in inhibition versus excitation or promoting release. Is this really dependent on the clamping domain, the accessory alpha helix?
So in Drosophila, we see roles for both the N-terminus and the C-terminus in clamping. Certainly the C-terminus prominently plays a role in localization of the protein, as you know. One of the isoforms in Drosophila has a CAA box that tethers it to the membrane. If you take that away, it completely prevents the protein from clamping. This isoform of it actually doesn't, has an amphipathic helix at the C-terminus, and that's where this biology is occurring. So it doesn't appear to affect snare binding at all. We've tested that. And the model that we have is affecting localization of the protein by changing potentially its interaction with lipid bilayers. The work in Complexin has sort of highlighted that. But I would say exactly how it unclamps release is unknown. What we do know is that the other mode of release of Complexin regulates is evoked responses. And as we have shown, and I think other people have shown, you can separate those two modes of release. So that the role of Complexin in clamping can be separated from its role in evoked release. And these mutations have no effect whatsoever on its role in evoked release. So they're specifically focused on its role in clamping.
Great. And a totally different question. Have you checked with this really fascinating observation of these different release probabilities of neighboring active zones? It would be interesting to know whether that's related to this regulation of the release probability via snare complex assembly by Monkey 13, in particular with all the DAG stuff.
Yeah, so in terms of sort of two questions there. One, do we see neighboring active zones having similar release probabilities? There's a little bit of evidence for that, but little. In terms of what else controls release probability, the ultimate goal that we want to do is to structurally reconstruct by serial section EM synapses that we've done release probability maps on, so we can define whether a high P active zone in fact has more docked vesicles than a low P active zone. And that would sort of give credence to the idea that there might be priming mechanisms that are differentially expressed there. But what we have to do is basically take our imaging approach into the various backgrounds, RIM, Monk 13, Monk 18, and begin to explore what really influences release probability and what doesn't. Or all of them decrease release probability, but what differentially affects it.
Troy, when you have this phosphomimetic in a fly, do you see differences in release, spontaneous release in the different release sites? And also, do you get a muscle cramping if you've got a lot of simultaneous?
Yep, so in terms of the phosphomimetic version, it basically increases release probability by about twofold, I think. Richard is in the audience somewhere, I assume, but it's about twofold, I think. Whereas the null mutation increases spontaneous release by 50-fold or more. So this phosphorylation does not completely inactivate the protein, that's for sure. It subtly modifies its function, but that's enough to give this transient increase in mini frequency.
Troy, this is beautiful. You can't apply this technology to some mammalian synapses, like the mossy-fiber synapse is pretty big.
Seems to be plenty of people working on mammalian synapses in the audience. I'm sure that so I think a more relevant question for us is, can we begin to apply this technology at synapses that are more plastic than the NMJ and the fly model, where we have genetic access? And so that's something, for example, in mushroom body neurons, that might be worth really looking at.
So Troy, it's really striking that synapse by synapse, you see no correlation, basically no correlation between calcium influx and release probability because, I mean, as you showed, if you change extracellular calcium, you do get an alteration. So are you sure that that's not maybe sort of a mixing bulk signal across synapses, something might be more related to facilitation than actually to release probability?
Well, I would say certainly for the synaptic vesicle release probability measurements, we have to do that with low stimulation frequency because we go to really high, we get overlapping calcium plumes that sort of muck everything up. So we're limited for postsynaptic release P measurements to relatively low stimulation frequency. For active zone calcium measurements, we typically do have to stimulate about 5 hertz to build up enough calcium to see. So again, whether that is reflective of what goes on during a single action potential, it's hard to say precisely, but I would say it's very consistent between different stimulation paradigms and that those individual sites show higher or lower levels than the average. So, you know, it's jumping between, but there is a correlation. I mean, there certainly is a correlation between calcium channel density and release probability. So if you look at, you know, maybe it wasn't quite as obvious and I underplayed it a little bit because I'm interested in some of the synaptic vesicle proteins and the role they play. But if you look at this data, for example, at some of the higher P sites, so these are, again, the high P sites, they typically do have higher calcium channel density. So some of these higher P sites do map to the sites, in fact, that have the highest calcium channel density. So it's not that there's no correlation, it's just that you don't get a perfect correlation when you really look at those measurements.
I guess you could try to calibrate it by changing calcium. Do a Regier style experiment, right? Change calcium, calibrate the whole thing, and then see whether the individual points are falling along that curve.
Yeah, we're certainly not quantitatively measuring presynaptic calcium. These are qualitative measurements for sure. I mean, GCAMs are not that great for quantitative. The beautiful thing, I mean, we were trying to get a red calcium sensor we could put in the postsynaptic cell and the green in the pre, but in our hands, the red calcium sensor is just not working.
So Troy, following up on this, it may be that you can't actually see nanodomain coupling because, you know, you're talking about a few nanometers that might be important here. So you might want to look at using something like BAPTA AM versus BAPTA EGTA sensitivity or something to get closer to that problem.
Yeah. The fly activosomes are around 200 nanometers, and we can see calcium influx to a single action potential. I don't want to say that we can't. We can, but it's only at the strongest synapses that we can see that. So with, you know, 1 hertz stimulation, you can see calcium signals presynaptically. The number of calcium channels that are there is, I think, like mammalian active zones, is pretty much a mystery, but, you know, 10 to 100 is sort of the predicted range. Somewhere in that ballpark is the idea.
Yeah, but it may not be the absolute calcium influx. It may be the relative localization of the channels to the actual.
That's right. That's right. That's right. We're tethering our sensor to BRP, which is a little bit away from the site of action. You're right.